Oh, mute.
Okay, well, I would like to introduce Dr. Alexandra Zimmer, and she is with OHSU. She's an associate professor of medicine, Division of Hematology, Medical Oncology, School of Medicine. Dr. Zimmer has lived and completed her training in Brazil, Europe, and the US. She specializes in breast cancer with an interest in better understanding and improving treatment for brain metastasis. She enjoys knowing her patients and their families and learning from them. Outside of work, Dr. Zimmer loves traveling, cycling, and hiking with her husband and son. She also enjoys gardening, reading, and singing. So I will turn it over to you, Dr. Zimmer. Thank you so much for being here. We're really excited for the presentation.
Thanks for having me here. I was asked to talk about clinical trials in breast cancer research, and there's a whole world to be talked about, but I kind of pinpoint some things. One idea was to get like 3 or 4 trials that I thought were interesting. First, as you saw, I've been working in clinical research since I trained in Brazil with some clinical trials there. And then I know this is not about me, but just so you know who I am, so you're not like, who is this woman talking about this? And then I went to actually live in Brussels, Europe, just to work with clinical trials in breast cancer, and then moved to the States and worked at NIH for a while before coming to the OHSU. I was focusing in several clinical trials in breast cancer, but mostly in brain metastases, which is my main interest. As you know, people kind of we cannot do everything. So that's one of my things. But coming here to the OHSU, we have a big group or big possibilities of research. And we are currently trying to get more colleagues, more breast oncologists to come to the area and work with us, developing new areas in research. When I was thinking about which trial, they asked me, well, talk about trials you think are interesting. I think I started to think about it, and I felt that, there's a lot of interesting things in different trials, depending on how you look at them. I was also thinking, well, maybe I should talk about trials that are here as well. And I think they are interesting because one thing is to talk about things that are not kind of available, because sometimes you have a very small trial in the corner of the US that only recruits patients for that area, and they nobody will like move there or things because of that, because of several reasons. And then I thought that why not talking about trials that we have available actually at OHSU and I think are very interesting. So I wanted to focus on that. And of course, if you have questions or other comments about that, please let me know. I can try to answer other things, and also suggestions for other talks also very welcome. I think information is key. People need to know about things so they can better judge and listen to recommendations. So I'm always happy to explain what I can. I want to this is a kind of a busy stuff, but I want us to focus on chatting on those things. Feel free to interrupt I know you have this system of like talking before and then asking later, but if you feel something is very hey, I cannot continue without that, let me know. So I don't know how familiar people are with treatments, with trials and things like that, or if someone was on or not on a trial. It all then the information is different, but I'll try to be very basic on things. And just to understand, I try to divide the trials not only in the subtypes of breast cancer that we know that we have the 3 main subtypes that are hormone positive, triple negative, and the HER2 positive ones. And so the treatment varies for those types of cancers. I try also to divide in different stages of disease, so different people can benefit from some information. And here we have, I will follow more or less the order here. I started with neoadjuvant trials for all subtypes, which means neoadjuvant, as some of you may know, is treatments that happen before the surgery, because the treatment of breast cancer involves, as you know, multidisciplinary treatments, and this is based on surgery, is based on systemic therapy, which is usually what the oncologist does with endocrine therapy or some sort of chemotherapy, immune therapy, or something like that. and then radiation oncology is part of that as well. The sequence of things can change a little bit, and we know we have changed a lot of them in the recent decades. And neoadjuvant are those, as I just was saying, that you do the treatment, systemic treatment before the surgery. So neoadjuvant, that's what we call. The main trial on that one, and I'm going to talk about that, is a broad trial for all subtypes of breast cancer that is called I-SPY. And I think it's a trial that changed the way we should think things. And I remember being in an early oncologist and hearing about this when I was in Brazil and all, and then coming here now, I'm able to work with that. It's amazing, and I just want to share with patients that it is some ways of thinking about treatment of disease. Adjuvant treatment is the one that happens after the surgery. And in this one, we have one, I want to comment on one for hormone positive disease that is HER2 negative, because specifically this is a trial that is dealing with ctDNA, with cell tumor free DNA. Which is something that a lot of patients have been hearing about and asking about. And I've been repeating to my patients that ask about that, what the ASCO societies and all, medical societies have recommended, that we should start looking at that in trials and not doing that sporadically, because there's a lot of information about this that we still don't know or how to use it. So I want to talk about that a little bit. And then metastatic breast cancer, and we have a specific one for triple negative here that I want to talk. And is a one that is developed and is available at OHSU specifically. So that's very interesting. And then brain metastasis is something that's my interest, and we developed some things at NIH when I was there, and now we are with NRG planning some trials to continue that, and certainly OHSU will be a center for this. I just want to plant that seed on there. It's not yet open, but something to think about. So I'll start moving over it, and you guys feel free to, just ask if something is very weird or needs more clarification. So this is the I-SPY that I mentioned before. It's a neoadjuvant, so before surgery. And this trial is very interesting in the sense that the main idea of the trial, the goal of the trial, is to see how much we can have a response of the tumors before the surgery. So we kind of in real time are proving that the breast cancer is responding to that treatment, because we are following with images and all. And as we know, and it's based on the idea or the proven concept that once you have treatments before surgery, you go to surgery and pathologists look at that material and cannot find tumor cells. That's what we call pathologic complete response. And when that happens is a good portender of better prognosis and less chance of the disease recur. Okay, so that's already proven to be a good thing. So we want, especially for HER2 positives and triple negative, we have that very clear. And the idea is, as we know, tumors are variable, even in the HER2, inside the HER2, inside the triple negatives, inside the hormone receptor positives. And sometimes we have a lot of treatments, a lot of drugs used, or very aggressive treatments that we do. We learn to do through the years. And for this last, I don't know, decades, we started questioning, like, do all patients need all this treatment? There are cases where we can maybe give better drugs if we have them available, or less drugs, and not give so much toxicity to the patients. And that's the main question of this trial, and the reason why, if possible, things can be evaluated before the surgery. So the idea of this trial, can you see my signal here, or should I use another? Can you see my arrow? You can? Okay. So so the idea of this trial is that people will, as usual in any trial, start screening consent, so we can check some things, and then the screening process. And then there is, depending on some results coming for this, that is basically an analysis of the size of the tumor, the presence of lymph node involvement, and the pathology of the tumor that is sent for a test that is similar to, maybe some of you guys heard about the Oncotype. There is a test called MammaPrint that is a little different and is not so widespreadly used here in the United States, because the oncotype is more specific for some things. But the mama print is more or less the same idea, that you look at the biopsy of the tumor, the tissue of the tumor, and have some ideas on prognostic and behavior and etc. And based on that, then there is a randomization based on what's the best possibility of treatment for these cases. And there is 3 blocks actually that the patient may or may not pass through. The first block, so the immediate way treatment that patients would receive, and that will depend. If you are hormone receptor positive, there is one group and several different drugs. If you are HER2 positive, then you go to another group of possibility of drugs. And if you are triple negative tumor, if you have a triple negative tumor, you go to another group that has other possibilities of drugs. And then, but the treatment A, or this initial one, is usually a novel targeted treatment, which is based on those results that we get from the analysis of the tumor. That is idea of testing new drugs that were evaluated before, have shown some potential treatment for these specific tumors, but still need to be proven in more patients, and more specifically if they can really get a good result. So the idea is we are giving something new here. And because that's new, that needs to be controlled very well, because we don't want to give a new thing and then let the disease go and people, not having a good response. So the initial evaluation is very controlled. People will receive this new drug, but in a short interval, like a maximum 3 weeks, they will have images again, usually an MRI, to look at that and see if there is some hint of response there. The treatment is continued with this drug. If there's no hint of response, or even in a little increase, then we will go to treatment B, okay? So if patients continue on treatment A, they are responding well, we continue following them for a next number of cycles, depending on what was planned for that drug, usually 4 or 6 cycles. And if the case is the tumor disappears or is very decreased on the images, we may go directly to surgery. That's the goal, okay? But again, these are new drugs. We can't promise that will happen. That's why we follow them so closely with the images. If the disease doesn't respond completely or is increasing or something, we go to the treatment B. And the treatment B is the one that is the standard for that type of tumor. So for HER2 positive will be some anti-HER2 agent with chemotherapy. For hormone receptor will be the specific hormone endocrine therapy. For triple negative, some chemotherapy. And this is the standard, so it's the usual thing that we do. We continue monitoring this with images. That becomes a little more like 6 weeks or 12 weeks, depending on the treatment and the timing. And then depending if there is still disease after that, there will be a discussion with the physician and the patient with those results, with the images and etc. And then we decide if we will add something else, which in this case could be some additional chemotherapy, for example, in the usual triple negatives and HER2, maybe, or even the hormone receptor positives, some doxorubicin or anthracycline that we know is one of the chemotherapies that we try to avoid as much as we can, because there may be heart toxicity or risk of leukemias and something in the future, which is low, but still we want to be away from that if we can. So that drug is reserved for this third option here. Or some immunotherapy, as we know, especially for triple negative. And then this finish the trial and we go for surgery. What is important to understand is that not everyone has to go through these 3 blocks. If you have a good response to the initial agent, it may be that you may go to surgery and then you don't need anything else. If you don't have a good response here, then you go to the standard treatment, which would be the treatment you would receive without a clinical trial. And then if still not perfect, we may have the option to offer something else. Not in all cases, but very potentially. So again, not everyone needs to go to all the 3, and it will depend on the response on the tumor and etc. that will be evaluated every time, okay? And then continues the follow-up. Points about this trial which are important is that it requires biopsies through the initial evaluations of the trial, so we can evaluate the responses, especially around the treatment A, which is a new treatment. And then because we want to be sure that there is response going on in the tumors, in the treatment, there is frequent MRIs. So people that don't tolerate MRIs and things like that would not be a good trial for them to be. Okay. So this is in a very broad thing, what is I-SPY. So for early stage disease before the surgery. Okay. Now, this is a little more complicated figure that I put here just to understand how much is involved on that. And it's not that patients should know about, I mean, patients should know about it, but there's not their responsibility. The team will be taking care of that. But just to show that because so many things are going around here, it may become, you know, as some trials go, a kind of busy trial in terms of requiring images every 3 weeks or 6 weeks or 12 weeks, biopsies in some points, and potential to go to different arms of treatment. But the goal is really to start with a new agent with less toxicity and potentially go to the surgery. If that's not seen, then we go to the standard or to a new treatment and go to the surgery. And more interesting, this trial is a trial that has been there for years now. There is distributed to the United States, so is available in West, East, North, South. It comes out for it started initially at San Francisco University, and it's now as again distributed to the country, so you can find it almost everywhere. And the drugs are changing. So as new drugs come to be available and developed, this arm may receive new drugs and then evaluate new drugs. And as we reach the point where the number necessary of patients to be evaluated to show some effect of the drug is there, this progresses to new trials, and then we receive new drugs, new possibilities of treatments here. So it's something that is evolving in itself. Okay. Now, for people that don't know about the what we call phase 1, 2, and 3 of the trial, sorry, I didn't say that in the beginning. I wanted to be clearer. This is a phase 2 trial, which is phase 1 trials are trials where we are testing absolutely new drugs just to see if they are, you know, not toxic, not too toxic. They are tolerable and then we start to see if there is possibility of responses. Phase 2 trials are trials where we know that drugs are tolerated, but we want to understand better if they are having the effect that we want for that specific tumor, which is exactly what I-SPY does because of this arm 1 where new drugs are tested. Then on the block 2, B and C, those are standard drugs. And then the phase 3 trials are trials where we're trying to compare treatments. So we have new treatments compared to standard. So it's usually the randomized ones where patients will receive one or other, and we want to understand how they do better in one or other. And that's how we have new treatments. Okay, this is a phase 2, so it's not a comparison. You will receive a new agent in the beginning, and then you go to a standard therapy if that new agent doesn't do everything that we expect in the beginning. All right. So key eligibility things, that those are very important because we need to understand who are the patients that can be in this trial. And I just highlighted here, this is the main and basic thing. If you have a new tumor of breast, independently if it is hormone receptor positive or HER2 or triple negative, the tumors that we want to, that are the best evaluable in these trials are tumors that are stage 2 or 3, because they have a size enough that we want to have a treatment before the surgery, and we can see the response in that. Other tumors are sometimes too small, and we cannot see very well the effect that we want to see in that. So just to be very clear, tumors, this stage 2 and 3 means exactly what is pointed out in these two dots here. If the lymph node is negative, tumors need to be bigger than 2.5 centimeters clinically, or more or equal to 2 centimeters on images. If the lymph node is positive, then the tumors can be a little smaller, not by much, but can be up to 2 centimeters or more in the clinical evaluation, or bigger than 1.5 in the images. Okay, that's the basic thing. Anyone that has any kind of subtype of breast cancer with these measurements or bigger would be someone considerable for these trials. And then there are the details of other things here that are, you know, you have, as I said, there's biopsies and there's MRI, so you must be able to tolerate that. Should not have previous surgeries to the lymph nodes in that side, because that affects the way that things are, the tumor behaves and how we can evaluate responses. And then as it should be patients, as I said, neoadjuvant. So you're coming for the initial treatment, and you have this initial treatment before the surgery. So previous treatments are not available, not good for this type of trial, because you already have an effect from other drugs, and it will, that makes it impossible for us to have a good evaluation here. And then again, no stage 4, because this is metastatic, and then we have other trials for that. No other, heart major problems and immune problems, and etc., and some arrhythmias here that these drugs this day are very involved with. But overall, is a very broad trial for neoadjuvant treatment, testing new drugs, potentially giving less treatment to patients, with a well-controlled setting of images and evaluation, so we can be sure that we are safe in not in offering better treatment when not causing problems to the patients. Okay. And through this treatment, I told you this trial has been there for years now. Lots of treatments that we know these days that are being tested in other trials, phase 3 and etc., came to be because of I-SPY, because they were tested this way. So this is very interesting, again, for early stage cases, and we have that available at OHSU. Now, I wanted to go to the one that I mentioned, that is the adjuvant trial. This is the only one I have here, which is early stage breast cancer and treatment given after the surgery. Okay, this is not the main treatment, let's say, and this is actually evaluating a tool of evaluation of patients more than in treatment as well, but it's built over the ctDNA. And I know you're going to have a talk or something in December about ctDNA, and I wanted just to put it there. We have this trial available at OHSU as well, and some other areas in the country. It's the only trial currently, bigger trial available that I know that is evaluating ctDNA. I'm hoping lots of trials are coming with that, because if you didn't, you will hear about ctDNA soon. This is a way that we look at blood work, and we look for DNA. Okay, there's tests, specifically tests looking for DNA, but not your DNA. We're looking for DNA that is strange to you, that is linked to the tumor. Okay, potentially to tumors, and in this case, breast cancer. This is a strategy that has been used, tested, and approved for colon cancer already. They have treatments related to the presence or not of this ctDNA and follow-up of patients. The problem in breast cancer is that though we found a test that can find ctDNA in this blood of patients with history of breast cancer, we still don't have any treatment linked to that. We don't know what to do with that after. Okay, so there is some trials that started to be presented in the recent years showing exactly that. They will follow patients that had breast cancer, not known to be metastatic. They just had the official treatment, surgery, chemo, or endocrine therapy, whatever. And at some point, people did this test, blood work, looked at the DNA, and found these days we have the Signatera. I think it's the most common one. And then they looked and found that there are tumor DNA there. And when they look to groups of patients that still are not big enough, but 70 patients or something like that, they found that majority of these patients, when that ctDNA is found there, despite not having any other evidence of disease, they are, if you follow them for a period, a long period or shorter, that's the point, one point. They don't know for how long, but majority of these patients will develop metastatic disease. So means that the cell is still there, despite of the treatments that were given. But not all patients developed metastatic disease. So this is a point that shows we still need to have it a little better. And also they don't know the time. We couldn't find a good time. Some patients develop disease years later, some patients develop months later, and we still didn't have a good way to understand who will be those patients. Moreover, when they found that, we still don't know what to do with that. Should we do images more frequently? Should we start some sort of the next line of treatment? What is the next line? And we don't know that as well. So it's still in the way I explain to my patients. I think this is the future. I think this will become standard of care, and we need to use this. But we still need to learn how to do that. And this is one trial that is doing that. Out of trials, my opinion and the opinion of the ASCO guidelines and major medical societies is this is not supposed to be done. Why? Because it will show you that there's the potential for your disease coming back. But I don't know if I should follow you with images every 3 months, every month, every year. I don't know if I should give you the next treatment or not. And remember, treatments have toxicities. And I don't even know if these treatments will be good enough. So trials need to be done to evaluate that exactly. And this is one of the trials that is doing that. So that's why I think it's so great. So this trial is a randomized phase 2 trial of circulating tumor DNA guided second-line adjuvant therapy. So what they want to is use exactly what it is. This is a tool. So let's use that. And if patients have that positive, we will change the treatment and see if that's effective or not. Okay. So for this trial, the idea was to have stage 2 or 3. Again, we just talked about in the other trial before, stage 2 or 3 are the patients that either have lymph node positive with any size of breast or tumor, or the ones that have lymph node negative breast cancer, but with a tumor that is about 2 centimeters or more. And in this case, they have to be hormone receptor positive because the treatment we are using is for those types. And are considering high risk patients, and I will tell you which ones they consider high risk. And the idea of this is that they would have a ctDNA test done, the Signatera, for example, as a screening, and then repeating every 6 months. If it's negative, continue that for every 6 months. If at some point becomes positive, or if it's positive by chance and the first time you check it, we do raise staging image, which usually would be CT scan and bone scan. And then if there's no evidence of metastatic disease, then the patients will be randomized, because we don't really know. I just told you that we don't know if changing treatments or all makes a difference. But here what they try to do is randomize. So one group of patients will continue exactly the adjuvant therapy that we usually do for endocrine disease. And the other group of patients will be escalated up, will receive the fulvestrant, which is usually a drug used for metastatic disease, and better working when you have resistance to some initial resistance to endocrine therapy, and then add a CDK4 like palbociclib for 2 years, okay, and see if that makes a difference. We continue following the ctDNA, and depending on how that is, it may become negative, it may become positive, that may continue to be positive. That's what the trial wants to understand. Can we impact that? Can we change that? So I think that's very interesting, and that's the only way I would be recommending to do ctDNA for the patients. Furthermore, what the eligibility, I know it's a lot of detail, but because it's a little complicated, I hope I can talk about them to you guys. But the idea is that you would recommend or do the ctDNA if you're thinking about this trial for your patients, in patients that have high risk for recurrence of the HER2 negative or hormone receptor positive breast cancer. And they may have completed endocrine therapy or are within the 7 years since the date of surgery. So it doesn't need to be immediately after you completed you just had the surgery, started endocrine therapy. It can be up to 7 years later. But no more than 7 years later, patients could still enter the trial, because it will be a follow-up. We'll do the ctDNA. Depending on the results, we continue just follow-up, or start another treatment, as I said, fulvestrant with palbociclib. Patients may have had CDK4 also in the adjuvant setting, because we know now that there were trials that evaluated the use of CDK for 2 years. One complication here is that some patients, now we have the Monarch E trial that is using abemaciclib in the initial 2 years post-surgery for hormone receptor positive patients that have high-risk disease. But I would understand that they would still be in this group as long as they are more than 12 months or more than a year after completing these 2 years of CDK4 as the initial treatment. High-risk disease is defined by this, and I'm going to tell you now, is the 4 or more lymph nodes that were positive when the surgery happened, or internal lymph node, and then or tumor size that was bigger than 5 centimeters, and at least one lymph node that was positive, or if patients that had diagnosis of inflammatory breast cancer, no prior fulvestrant, etc. I think this one is a little more complicated, but usually inflammatory breast cancer means chemotherapy, so that those patients will be a little different than these, but they could potentially be enrolled in the trial. So that's what they characterize as high risk, is a lot of lymph nodes or a big tumor that has one lymph node. And those are the patients that will be the ones enrolling in this trial. I anticipate that in the future and soon we will have trials that will have patients with not so high risk as well. But this is the way that we try to see a signal for some sort of response and have some evidence in the trials, you know. So that's the main thing. And again, I wanted to mention this one because I think it's an interesting way, and it's the way that we should be using ctDNA in the future. Now, this is the trial for the triple negative patients that have metastatic disease. So now we narrow to a subtype again, a triple negative, which is a type of tumor that we know that some patients do very well with the chemotherapy, but some patients, unfortunately, will have that aggressive form of disease where we can hardly find a good treatment and becomes metastatic, and we kind of try to find new ways. And we are finding new drugs, but there are still so much to be done. And this is a trial that is, again, a phase 2 that is, there are several drugs here. It doesn't mean that people will receive all drugs. It just means that we will try to personalize that treatment to these patients. So the idea, and this is a trial that is at OHSU only, because it's been developed here with all the science background that we have and the possibility for testing that we have here. This is, again, triple negative patients, which means that they will have estrogen receptor less than 10%, progesterone less than 10%, and HER2 that is negative, non-amplified. One exclusion, and that's why we need to have an initial, for eligibility, we need to have an initial evaluation of the tissue of the tumor to be sure that there's no androgen receptor in these tumors, that is, and if there is if it's not bigger than 80%. Because this type of the triple negative tend to have a different behavior and potentially not benefit from these drugs here. So that's why we need to exclude that. It's not super common, but it's something necessary so we can offer, we may be sure that we are not giving the wrong treatment potentially to these patients. So patients can be in the trial as if they had already 2 lines of chemo, up to 2 lines of chemotherapy before, in the metastatic setting of triple negative. But they cannot have received a PARP inhibitor, which is kind of olaparib and other agents, because we will be using those treatments in the trial. As I just said, patients can sign the consent and enroll in the trial, but we just need to have that initial evaluation of the tissue, maybe from a previous biopsy, or if there is no tissue enough, or we don't have any result recently, may need an initial biopsy to understand if it is proof that is triple negative, and especially that is no androgen receptor bigger than 80%. So involves an initial biopsy in the trial. Then we start, so we don't delay treatments. We start with a olaparib agent, which is a PARP inhibitor that tends to be effective in several different tumors, especially BRCA positive patients, BRCA mutations, but also has demonstrated some potential effect in triple negatives. And then we use that for 28 days, every patient that enters the trial, and have a repeat biopsy. And then we can evaluate some changes that may happen in the tumor. Why is that important? Because depending on the changes, we'll look at several different pathways of mutations that there are in those tumors. And depending on the positivity or not and signals that come there, patients may be randomized to one of 4 arms. So you go to a different possibilities, and based on the tumor that you have there, based on the patient-specific tumor, we will try to tailor the treatment specifically. And for now, we have these 4 arms here, all 3 of them containing the olaparib that was received already, and combining a new drug. This first arm is combining durvalumab, which is immune therapy agent. But require a certain mutations to be there. If those are not there, then we go to evaluation for tumor for arm 2, which is offering olaparib, that was the same drug here, but plus the selumetinib, which is a MEK inhibitor, which is an inhibitor of this pathway that is called the MEK, and is present in some tumors. If this is not still the one for that patient based on that biopsy result, we'll go for the third arm, that is olaparib and capivasertib, which is an AKT inhibitor. Again, will inhibit that kind of pathway through the AKT way. And then if again, not even this mutation is present, we have the fourth arm, which is giving the ceralasertib alone, which is an agent that is an ATR inhibitor. Okay, those agents have their side effects. So I was explaining to a patient this other day, they are very targeted agents to specific mutations, and they cannot be called chemotherapy per se, but they also have side effects, and depending on the arm, those will be a little one different than the other. So in those cases, we should evaluate. But the idea is to see how the response goes, evaluating images and etc. in these patients. There's the option for a third biopsy on if progression happens. Other than that, patients should continue on treatment. And then while they are responding or while there is no toxicity that is imperative, okay? As I said, just to understand how this decision tree goes, I told you that there is a biopsy and then you can go to one of each 4 arms. The decision tree is more or less like this. If your tumor have high mutation burden or PDL more than 1%, or on treatment basal-like immune activated, so specific things that are evaluated on the pathology of tumor, then patients will be selected to receive this first arm, that is olaparib and durvalumab, that is immune therapy. If that is not there, then there's another level of tests that go on. And this happens at the same time, okay? It's not like we will be waiting. We'll do all, and then we can explain to you why you will be going to receive one or other treatment based on specific mutations that we're going to see. Okay, so the main inclusion criteria for these are, again, to be a triple negative, and this is the usual thing, that is estrogen receptor and progesterone receptor less than 10%, and HER2 non-amplified. Again, we need the androgen receptor evaluation on the beginning. And then we need to have a measurable site of disease, and that's been one problem occasionally in some patients, that we cannot find the measurable thing because there is pleural effusion, or there is some sort of effusion that's the problem, or some bone lesion that we cannot evaluate. But we need to have something measurable that we can understand that the treatment is being effective. So that's one thing that sometimes is a problem. Patients with BRCA mutation are eligible for the treatment, and again, up to 2 previous lines of therapy, chemotherapy in metastatic disease are allowed as well. Okay. And then exclusion criteria is if you have a bad immune disease that we cannot give one of the immune therapies. If you have serious infections like hepatitis and HIV that are not controlled and can be affected by these treatments, unfortunately. So that is not possible. Patients that have brain metastasis can enter the trial unless they have symptomatic disease that needs specific treatments and may delay things, or we could cause more problem to them. And then there, depending on the drugs and the arms, there may be specific exclusion because of the specific treatments, but that will depend on the treatment itself. So again, this is for triple negative metastatic disease, available at OHSU only, and a tailored treatment specific to the tumor that the patient has. So patients with metastatic triple negative that have had up to 2 chemotherapy lines in the metastatic setting would be available for this. We need to evaluate the initial tumor, and that may delay a little bit the enrollment. So anyone that is thinking about that, the sooner the better. And then I want to go to another, and this is things that are very close to my heart, because those are trials that I developed and are related to brain metastases. And I know that's a specific group of patients, but we know that triple negative and HER2 positive are very, very linked to the development of brain metastases. If you have metastatic triple negative or HER2, the chances of developing brain metastases are about 50%. So we did this trial at NIH where we evaluated we found actually researchers at NIH found that I worked with from the lab. They found that temozolomide, which is a drug that is used for brain tumors, not so shown to be effective for breast tumors, but because there is penetrance in the brain, Dr. Patricia Stig, which is a investigator from NIH, decided to try that in mice models of brain metastasis from breast cancer and see if using that continuously, it would avoid the development of brain metastasis in these mice that otherwise would have developed brain metastasis. And what she found was, and her group found, was that given this temozolomide every day, small doses, these mice will not develop any brain metastasis, despite having the chance being built for that, as opposed to the other ones where they gave control things, some sort of, saline or something, they would develop. So there was a lot of problem with the model. They were developing brain metastasis, but the temozolomide controlled that, and they didn't have any brain metastasis. When I started working at NIH, I looked at that and I said, why this is not tried in patients? And there are several issues. One of the things is that because we don't know, though we know HER2 and triple negative have a high chance of developing brain metastasis, we don't know more than that. And we know there's a lot of them that will not develop brain metastasis. So you can imagine the cost of a trial and using a drug that can potentially have side effects in everyone that has HER2 or triple negative. That's not reasonable. So we were trying to understand who are these patients that will be a chance to be preventing new metastasis. And the idea was, if we have patients that already developed brain metastasis and are HER2 positive disease, we started with that one because there's more treatment. We know more about the treatment. So if patients had brain metastasis, received local radiation to this brain metastasis, we know that they will be at higher chance of developing new brain metastasis still. Actually, there is data showing that if patients receive radiation locally, stereotactic radiation to brain metastasis, in 1 year, very likely half of them will have developed new brain metastasis already. So looking into that, our idea was to give temozolomide to these patients after they got the radiation for this initial brain metastasis, and add a drug that will be otherwise the treatment for their cancer, breast cancer, because temozolomide doesn't treat breast cancer. There's something that it does there in the brain that avoids these metastases, but it doesn't treat well breast cancer, not as other drugs. So in HER2, we propose to use the TDM1 or the Kadcyla to treat the systemic disease, and then add the TDM1 to prevent new brain metastases. And because we didn't know if it was safe, we had to do a phase 1 trial to be sure of that first. And this trial was completed already. And actually what we noticed is that patients that we had 12 patients enrolled with different doses of temozolomide. All of them received TDM1 or Enhertu or Kadcyla. And actually, at least there were only 2 patients that developed new brain metastasis. All the other 10 didn't develop brain metastasis. And that was, these 2 patients actually was one patient that developed very early in the first month. So probably metastasis was already there. And the other one was someone that had already 20 metastases in the brain. So really a very advanced disease. But all of these patients have a good benefit, and most of them went over 1 year without new brain metastasis. So there's something there. And we need now to expand that to more patients. Now, we acknowledge that there are new treatments beside the Kadcyla. We have the Enhertu that is doing a lot for patients with HER2 positive disease. So the new trial that we are proposing is actually this one that is written phase 2 here, which is kind of the same group of patients, but instead of receiving TDM1 and temozolomide, they would receive TDM1 or trastuzumab deruxtecan, which is the Enhertu, depending on what they already had before, plus the temozolomide, which is given every day as an oral drug. But we had to randomize then patients to, and I know patients are really not good fans of randomizations because you may enter the arm that you don't like, but we didn't find a better way to look at that yet to prove things, right? Because it may as well be that it doesn't make any difference if you use temozolomide. We need to use it in a bigger group, but we need to have a comparison group to make sure it makes the difference. So this is the trial that we are trying to do. And here I want to just to raise one thing about trials and how important groups like yours are, patients and patient advocates. Because if you, temozolomide is a drug that actually is an old drug. It was produced by Merck, and one of the big pharmaceutical companies, and they have developed this drug for a long time. And after some long period, these drugs go off patent, so anyone can produce those drugs, and the companies don't get so much money around them. So they don't have interest in doing that. When I did this phase 1 trial at NIH, they still were around that time that will become off patent, and they paid for the temozolomide for our patients in the trial. The drugs that we gave were actually the other drugs for treatment of breast cancer were standard, so no problem with that. The insurance will pay. But now for the phase 2, they said, well, we don't gain any money with that. We don't have interest in this, so we are not paying for it. And it's not a standard drug. So our big problem to develop this trial has been to find a way to get the drug, to get the temozolomide and pay for it. So we are talking to some companies. We have some advocate groups that, including one patient of mine that was in this trial that wants to have other patients involved as well. She is working with advocacy and has been very vocal trying to get government and all involved on that. It's when patients can say, hey, this is important for us, not the companies that are getting the money and all that. So I'm just mentioning this to you just to say how important it is when patients know how things are going. And this trial, by the way, was developed always with participation of patients. Since the beginning of this trial, when we start thinking about it, we get groups. It was developed at the East Coast, so we had groups from there that came to our meetings. We discussed exactly as I'm talking to you today, what we knew, what we thought, and how we thought things should be done. And they were saying, well, and we will ask, so we had actually lumbar punctures 2 times in this trial, just to see if we could find some marker that will tell us the disease was responding, or we could select patients in the future to receive the treatment. We found very interesting things that I'm not going to go over now, but I can talk specifically about brain metastasis at some point. And the patients, I remember one of the big questions to them was, because we were developing the trial, a lot of people will say, patients will not accept lumbar puncture. They will not get it. You cannot do this. And then we had these patients advocacy groups working with us, and we posed the question, hey, the goal to do that is this and this and this, and that's what we would like to propose. And they told us, yeah, it's not fun, but we want to have these things done better. We want improvement, and if that's what it takes, we are okay doing it. And we were able to do the lumbar puncture, and now we found very interesting things that will probably involve some evolution in the future, and that was only because these patients were there rallying for it and helping to disseminate the word, even when physicians sometimes think they know better than the trials and all that. So I remember when I talked to Susan Stearns, that is there, I see her, that one experience that I had for a long time is that of working with patients in development of trials. And that's one passion that I have, that any trial that we think about, we should have patients sitting with us and talking about how does that feel to you, and not because, personal and isolated opinions are good, but you have experience of talking with your friends and people that have lived with breast cancer and all, and you can transmit to the physicians, the broad feeling. And sometimes it's not that we are I don't want to do that thing about, well, the physicians don't understand anything and want to do this, whatever. It's not like that. We are trying to get close, but you never know a disease until you live with it, right? So it's good to have that approach and understand, hey, that's how we think about this disease and how we know medicine and the things about it, but it affects your life. So if you can tell me how we can get to a point where it's acceptable to you and where you see that makes sense, that conversation I think is very interesting, as well as the conversation with people from the lab, right? Because we are the clinicians working with things with patients. They are in the lab, and sometimes they don't understand the implications of that in real life, because they are working with mice and looking at pathways and things, not necessarily seeing how that actually in the real life of patients will affect several other points. And as well, we don't understand sometimes those pathways. So I think communication between all the steps, it's so important to develop those things. So if I can leave a message here, would be that I know trials are not perfect. It's the best thing we can do, but I think we can do better if everyone talks about it, compromises a little bit. And I hope that as we evolve at research and breast cancer research at OHSU, we can get an open communication and a participation of your group, other groups, in the development of those things. So with that, I think that's and I just wanted to show the last slide. If anyone doesn't know, which I think everyone does, this is the place where physicians go looking for trials, okay? So if I'm looking for a trial, that's where I go. It may be overwhelming because you will find every little thing, and sometimes they are working in making it very very, very current. But sometimes there are things changing there. So, if my patients bring a list of that, and to me, I'll be happy to look at it, maybe not in the same day, but to understand, things, because we are sometimes not, up to all the things that are there. But the trials should be there. There are groups that have lists of trials specific, like the metastatic breast cancer group and things like that, which is very useful because then already they have a review and some more data, more discussion about it. But I would say that's the main source for trials. Everyone that does a trial has to register with the clinicaltrials.gov. So every good trial should be there. And I would say that's a start. Certainly not the end of the search, but it's a possibility. So with that, that's my last slide. And as you see, I can talk a lot. So if you have any comments, questions, or things, I'm happy to try to answer.
This is Linda. I have kind of a general question. Where do you find some of the most typical support or financial support for clinical trials?
Yeah, so those things we have big funding places that are the NIH is one big funding place.
Sure.
Then we have several grants that are the big grants like DOD and other things like that. And then we have the companies that do that, and then the patient groups that do the through several donations. Not, I would say it shouldn't be from the patients, but because you're working onto that, get big groups to give these donations. We could make a list and provide you guys with that. I have groups that have those things ready. From the top of my head, is hard to.
Thank you.
Mm-hmm.
Well, I have a question that was sent to me.
Mm-hmm.
So the question is, any trials for metastatic but HR positive and HER2 negative going on or coming up soon?
Oh, there are a lot of them. We could talk about them just specifically. There are different ways to use CDK, different ways to add other drugs to that. We're gonna go to San Antonio. Probably will have a lot in December. We have this big meeting of breast cancer in San Antonio, Texas, where we usually get reports and updates and proposals of new trials. So we could, that's one source for looking. I'm sure around that time, early December, news will pop everywhere related to breast cancer about things coming up and all that. And I'm happy to talk again about it and do specific trials for each subtype.
Thank you.
Hi, Dr. Zimmer.
Hey.
Thank you so much for being here. This is a phenomenal amount of content.
I know. I'm sorry to overwhelm everyone, but
it's good for us, and I'm glad this is recorded because we all probably need to watch this 2 or 3 times for more of it to soak in. I'm going to repeat a question that came up at our recent metastatic breast cancer dinner series event, that luncheon. But just really for patients, I'll say who are not yours, what advice do you have for how they should approach their doctor, and not that you need to name other systems in the area, but just advice for the average patient who is interested in exploring trial options, and what they can do, and then what they should ask their doctor to do, and again, just general information about how patients can learn more if they actually want to participate and to make a difference in their life.
I think the main question is always that, is there a trial available for me? That's the broad question, and people may, just say, I don't know, whatever. But that's the main question. And if there is trials, is there any place here around us that you would recommend for me to ask for trials? And then probably they would, if it's someone, I don't know, I'm still learning about Portland and all these aspects, but if they don't have a trial, they will tell you, I don't know, OHSU or this or that, look it up. And I have to say that we have been a difficult thing, OHSU per se, because since I came, I'm the only breast oncologist. So we kind of, like, hey, we cannot see you, but we are opening. We have someone now, and I'm telling you this because I want you to spread the word. We have someone that is doing locums for us for 6 months, and we are hiring new people that are interviewing at this moment. But with these locums, I'll be able to see more patients. And so we intend to be more open to seeing patients and second opinions and things like that. One thing that we kept open throughout this time has been for clinical trials. So we always say, if you have a patient for clinical trials, send to us. We will review if they are eligible or not. It may be that already from the get-go, we know that they are not. So that's let's not waste time for any of us, including the patients. And then we go from there. Okay, I actually had to shut a little bit of my second opinion clinic and all that, but we plan to be opening that soon. I know other places have that as well. I tell my patients they should always get a second opinion if they feel that that's helpful and makes them feel better, because frequently you will hear a little bit of different things, because not everyone sees the same way, but they should be more or less the same kind of idea. If that's too different, then someone is crazy. But at least you feel more comfortable. And but again, through the trials, I would say ask if there's a clinical trial available to you, because there are patients that are not interested in that, really, because as I just described to this, all that involves a lot of participation tests and things like that, and accepting what is given to you, because it's not like once you randomized, you go there. There's not like, hey, I don't want this, then you're off. But because there's the only way we can evaluate as much as we know. But for the ones that are interested, I would say first thing is ask your doctor and ask where could I go? I like you, I appreciate your work, but if I want a trial, where could I go to hear about it? And then the other thing is go to these sites and I know that involves much more work, but if you at least find one or two that you find interesting, you can see where they are done, because that's one thing that happens in these pages. They tell you this trial is open. If you go down, scroll down these pages about the trials, they say where the trials are open. And then you can see if there's one close to you, and you can ask directly, or even I had patients that brought me pages of trials that they wanted me to, and I may not know about that, and I will look up, and I may look at it immediately, say, hey, but because of this, you're not, or this is not good, or this is good, you should, or it's done only in the other corner. So, but I think the first thing would be I will ask my doctor about possibilities around me. And then the other thing, look up, starting for the clinicaltrials.gov, that can be overwhelming, I understand. So the best thing after that will be getting one of those groups of metastatic breast cancer and things where they select trials that people looked before and think are interesting, and then show to your doctor or look directly. I had lots of patients that emailed me directly when I was at NIH about, and even now, that they saw a trial on this clinicaltrials.gov or somewhere else, and they wanted to know about it. And then we put our team immediately in contact because we want to have patients on the trials as well. And then it may not be the doctor directly, but the team will get in touch and understand if that's possible to see, and see if you're available, if you're eligible or not.
Well, I have a basic question. If you're in one healthcare system, but you want to participate in like an OHSU trial, but you're not through OHSU,
No,
is that?
that should be fine. People should, you have to transfer your care for a little bit during the clinical trial, right? But after you're completed, you can go back.
Okay.
And sometimes we try to do, for example, at OHSU, we have the community sites where people can have some of the trials. But yeah, usually depending on where the trials are open, you will have to go there for that period and then go back to your primary oncologist. We used to do that a lot at NIH, for example.
Gotcha. Okay. Thank you for clearing that up for me. I was always
Yeah.
wondering about that.
Yeah.
Sorry, one more, and this is actually clarifying Linda's question. One part of that is who is funding trials, but the other side of that is what financial support might there be for patient if they enroll in a trial.
Oh, yeah.
So if you can clarify that, the second part.
Yeah. So depending on it depends on the trial. Okay. Some trials already are built with support for, traveling, where the patients can stay, things like that. The trial one thing of trials, is that all your images, all things involving trials in terms of images, tests, and etc., it should be paid by the trial. Okay. That's a given. Or your insurance or whatever. But there's certainly extra cost if you have to be traveling there all the time, and if you have to be going to another city, sometimes, staying or something like that. And some trials build that up in them. We try to discuss when we discuss with pharmaceutical companies or the big developers of trials. Not always we are able to have that, but we try to. So that becomes a kind of a specific thing in every trial that you need to look for.
Thanks so much.
Mm-hmm.
Well, I don't know if there's any other questions. I do also appreciate your reminding us how important patient advocacy is in the clinical trials world and just in general. So thank you for that. Really
Yeah,
great.
no, I want the group to be very aware of this, and hopefully we can start some sort of work in that sense, because that is very important, All right. So I really appreciate being here with you guys. The ones who know me know that I love talking about these things, so I can talk forever.
Great, your passion shows.
You have to stop me. But whatever, whenever you guys have some sort of, brief moments, big moments, whatever, happy to chat with you, clarify things. Again, what I think people, knowledge is the key to everything. We will never be able to know everything, but if people can explain to us where they are coming from and we can hear some of that, we can, improve several things. So thanks for the opportunity, and I hope we have several in the future, and I'm here for whatever you need.
Thank you so much. We really, truly appreciate that. So
All right.
Thank you, Dr. Zimmer, and we will see you
Bye.
all later. Have a good night, everyone.
You too.